The nose swab's journey seems pretty direct, going from here to here to here. But before it ends here, its journey may continue as part of a mapping of genomic sequences. It's being used by scientists all around the world to track the emergence of SARS-CoV-2 variants. Here's how it works. Remember that nose swab you took? If it was a PCR test, around 5 to 10% of them end up here, in laboratories set up to conduct genomic sequencing. Genomic sequencing is a process used to analyze the genetic makeup of viruses.
It's sort of like creating and then assembling really large puzzles. The SARS-CoV-2 genome is about 30,000 bases long. It's an RNA, single-stranded positive RNA virus. And that length, if you just typed out the A's, U's, and G's and C's, is about the same length as the U.S. Constitution and Bill of Rights.
All viruses, like SARS-CoV-2, continually evolve as they replicate. Those changes in the genetic code are called mutations. A variant has one or more of these mutations. Labs continuously analyze the genome of SARS-CoV-2 to keep track of variants that pose threats to public health.
A lot of the laboratory testing itself is, moving a lot of tubes around. The example of contagion gives you a good sense of what that process kind of looks like on the back end.
Blue is virus and the gold is human and the red is the viral attachment protein.
We have tools like NEXTSTRAIN and MICROREACT and others that allow us to put sequence data into place and time. For SARS-CoV-2, most labs use an approach called AMPLICON sequencing, which means that small overlapping pieces of the genome are amplified by PCR, same sort of approach that is used for testing. They're sequenced, and then powerful bioinformatic pipeline and algorithms are used to then put those pieces back together and compare them to other genomes.
The sequencing is done by a mix of academic and clinical laboratories, along with local, state, and federal agencies. Within 2 weeks from this sensation, the sequence is available in public databases. While lab results will tell you if you're positive or negative, they most likely won't tell you what variant you had. But is there any way to find out?
I think you can probably make an educated guess. If you were infected over the summer and fall in the United States, the high likelihood, just based on the viruses that were circulating, is that you'd have a Delta virus.
Genetic sequencing often focuses on the spike protein, since it's the tool the virus uses to penetrate cells and spread infection. Genomic sequencing monitors mutations and allows scientists to assess how evolving variants might impact existing treatments. If a variant emerges, it's classified by the CDC into 4 categories: variant being monitored, variant of interest, variant of concern, and variant of high consequence. Let's take a look at OMICRON, which falls into the variant of concern category.
Omicron does look pretty different from both Delta as well as many of the viruses that went before it. It was first identified by public health research groups in Botswana and South Africa, specifically Dr. Moyo and the team at the Botswana-Harvard AIDS Institute partnership. It has an accumulation of 32 documented mutations in the spike protein, which sounds like a lot because it is. It has that deletion at residues 69 and 70, which lead to that SGTF or S gene target failure marker that is often used to understand where and how it's transmitted. That was important with some of the early cases of Omicron, mostly because a lot of what we've been seeing in the United States over the course of the summer and early fall was Delta, which lacked that mutation. So when you started to see an increasing number of these SGTF failures, it gave you a good indication of how much Omicron you're seeing across your sample, even when you didn't have the sequencing data in hand.
With variants of concern, the existing therapeutics, vaccines, and treatments all still work to stop the virus's spread, but it may not be as effective. The next category, variants of high consequence, is the most serious.
Now, we haven't seen a variant of high consequence yet. Omicron is much more transmissible than other reported variants. It shows evidence of immune evasion, even among vaccinated individuals, and it's hitting many communities and healthcare systems hard. The specific risk categorization, though, shouldn't mean that we treat any of these viruses with any less seriousness.
The emergence of new variants also begs the question, what should we call them? Well, the answer depends on who you ask.
The PANGO nomenclature system was like B117. There are NEXTSTRAIN nomenclatures that are more like a named hurricane type approach. So those would be like 21A or 21B. They have usually a year and then a letter. All of these can be a mouthful. And so the WHO, over the course of last spring and summer, brought together a group to help name and designate, come up with a consistent nomenclature system for variants of interest and variants of concern. The names have to be specific, they have to be distinctive, they have to be easy to pronounce and search for in multiple languages, and most importantly, they shouldn't be associated with a specific place or people. If you think of viruses like Marburg from Germany, Norwalk from Ohio, all of those are viruses that are associated with the places where they were first identified, sometimes pretty negatively.
So the WHO decided on Greek letters. It's hard to imagine the course of this pandemic without genomic sequencing. Even if you've never heard of it before, it's played an important role in monitoring and adapting to the pandemic's global scale.
I think one of the most important lessons, at least from the point of view of genomic surveillance, is that open data and rapid data sharing is an incredibly useful tool for public health and pandemic response. The potential for global public health is really exciting, but there are a lot of barriers that we have to overcome. How do we pull all these different types of data together? How do we actually collect the samples that we need to collect in a timely way so that we can get the information that we need? When you go to a test site and get your nose swab, you're really helping to understand transmission dynamics. So that'll help us understand not only the virus that infected you personally, but also trends in your local region, in your state, and across the country.